All right, so this is chapter 7, and we are looking at membranes, membrane structure and their functions. I'll be looking at some transportation mechanisms that happen in the on the membrane in cells generally. Okay. Now, from chapter 6, I told you that cell membrane is semi-permeable and that it is made up of both hydrophilic head and hydrophobic tail. The phosphate group that make the phosphate that makes the head of cell membrane, the head, it's hydrophilic, and the two fatty acid that makes the tail, they are hydrophobic. And I told you that any structure that have both hydrophobic and hydrophilic portion, that structure is called amphipathic structure. Any structure that have both hydrophilic and hydrophobic region is called amphipathic structure. Now, so when you look at the phospholipid membrane, this is what we call the phospholipid bilayer rather. This is what we call phospholipid. This is a phosphate head, and here there are two fatty acid tails. So every phosphate have two fatty acid attached to it. So that is the way it is. And you see, this is the first layer and then the second layer. And I told you the purpose of these layers is simply because there's water in the cytoplasm and also water in the extracellular environment. So because of that, the cell membrane needs to be protected, because now we said that the fatty acid, all of this region is hydrophobic. Hydrophobic means that they are fearful of water and they cannot relate with water. So the reason the cell membrane is actually double membrane is for adaptation mechanism. It's so that the hydrophobic region is not touching water. That is why it's double membrane. It's to prevent water from accessing the fatty acid area. That is the purpose of the double membrane. Now, we say the fluid mosaic model. The fluid mosaic model simply means that now you see you have proteins that are protein molecules bubbling in fluid. That is why we call it this fluid mosaic model. So it's of the fluid mosaic membrane of a model structure. You see, it depicts the membrane as a mosaic of protein. The further protein is embedded in the cell membrane within the cholesterol region, where it's able to move around. That's what we call fluid mosaic model. And we'll talk about its implication over time as we go. All right. Now, so membranes are held together mainly by weak hydrophobic interaction, right? The most of the lipids and some proteins can move sideways within them, which is why it's called fluid mosaic. The protein, they are not just static, they are able to move. Really, a lipid may flip-flop across the membrane from one phospholipid to the other. Let's go. Let's go. Now, membrane fluidity. Now, key thing about the cell membrane that makes it unique is that as temperature cools, the membrane can switch from fluid state to solid. And I'll tell you why. Then when temperature also rises, it can also move from solid to liquid. The liquid state is what allow the proteins that are embedded in the cell membrane to move, and that's why we call it fluid mosaic. All right. Now, the membranes that are rich in unsaturated fatty acid are more fluid than those rich in saturated fatty acid. Now, look at this here. Now, what is saturated fatty acid? What is unsaturated fatty acid? Saturated fatty acid are the acid that have, they are the fatty acids that have if you remember now from chapter 4 and 5, you remember that we talk about lipids, and I talk about saturated fatty acid. Saturated fatty acid means that it is saturated with hydrogen. It means there's no double bonds there. When you are looking at the fatty acid, you only have single bond all across. That is a saturated fatty acid. That is the kind of fatty acid or the kind of fats that you see in animals. That is the kind of fat that you see in animal source. They become solidified at room temperature. And then you have unsaturated in plants, which is why you see the oil that come from plants are not solidified at room temperature. That is saturated, unsaturated. Unsaturated will mean that it has single bonds. It has double bonds and less hydrogen. So if you look at this, of course, this is unsaturated. You can see it's not packed up, whereas this is saturated. This will be difficult to digest than this. This is going to appear like fluid. This is going to be like viscous. This will make it difficult for protein to move around it. This makes it easy for protein to move around it. All right. And the fact that the membrane is fluid make it easy for the membrane to work the right way it should work and be efficient. All right. Now, what is that? What makes the fluid, what we are talking about, solidified lipid or so in the membrane? It is cholesterol. So cholesterol is what we have in the cell membrane, and it's able to, at room temperature, it will restrain movement. At warm temperature rather, is going to, you see cholesterol restrain movement of phospholipid, but at cool temperature, it maintains fluidity by preventing tight packing at such point. So plants use different but quite related steroids, lipid to bother membrane fluidity. Let's go. I'm just going to run through on things that I feel you should not focus on too much. All right, we've talked about this. All right, so the lipid composition of the environment will be adapted to, of the cell membrane will be adapted based on the environmental condition, right? So for example, cell membrane have a high proportion of unsaturated hydrocarbon tails in fish that live in extreme cold. Does that make sense? So if fish that live in extreme cold, because the temperature in the area is cold, they need to have high level of unsaturated hydrocarbon so that there's no risk of freezing and the cholesterol is not going to be solidified. The hydrocarbon is not going to be solidified. The organisms living in variable conditions are able to change lipid composition in response to temperature that changes in the environment, just like us. For example, in winter wheat, the percentage of unsaturated phospholipid increases in autumn to prevent membrane solidification during winter. All right, so a membrane is a collage of protein. So on the cell membrane, you have protein there, and they are usually clustered in groups. We talk about we talk about peripheral protein, we talk about integral protein that are embedded in the membrane. And obviously, the purpose of the protein is for transportation, is for cell identification, for cell communication generally. As well, I told you the major determinant of the functions that your body perform, that my body perform, are the proteins. So the proteins are the one that determine what happened in our body. So it is expedient to have them there. The protein are the one that will serve as receptor that determines the activity of hormones. The protein determines the activity of different cascade effects that happen on the cell. The protein composition of the membrane may vary depending on the cell and depending on the kind of organism that we have. Again, I've just talked about peripheral protein, and I've touched this in chapter 5, so in chapter 6 rather, and that's why I'm just moving past it. So we talk about peripheral protein, which are proteins that are on the surface. Then we have integral protein that span through the entire membrane. All right, transmembrane proteins are integral protein. You see, that's what I just mentioned, that span through the entire membrane. Hydrophobic region of an integral protein will consist of non-polar amino acid so that it can still orientate with it and be functional. All right, now major function that protein does in our body are these: they help with transportation, they help with enzymatic activities, they help to signal for signal transduction, cell-to-cell recognition, intercellular joining. We talked about intercellular joining. Oh, sorry, I didn't talk about it with you. It was my AP student. So, when you talk about intercellular joining, you're talking about cell junction. Maybe I would have mentioned in chapter 6, cell junctions. Cell junctions is what connects cells together so that they can form tissues. So, you have a group of cells that work together to perform a specific function. It's called tissues. So, intercellular joining is when the cells join together, and there are proteins that join them together. Like we talk about one of them, coercin. We talk about cell adhesion molecules, CAM. Right? So those are the functions that protein does predominantly in our body. Right? I'm just going to continue to the portion. All right. Now we have some glucose in our system, in our cell membrane as well, that are coupled with lipids. So we call it glycolipids. All right. So when some carbohydrates are bonded with lipids, we call it glycolipids. And when some are bonded to protein, we call it glycoproteins. Now, majorly, they also enables the diversity of surface carbohydrates also help the protein and encode the cell membrane to function the way it should function. It helps with markers. Now, there are proteins on your cell membrane that are like markers. They are like markers. They help to identify. It's just like sticker that helps to tell this is this person's office. This is what this person is. So there are some proteins also on the cell that helps to determine what kind of cell is this. So when there's a need now, let's say an hormone is sent out through the blood, the hormone knows where it's going to because there's a protein that is there as a receptor. All right. So there are proteins that help with cell identification, cell recognition as well, like that. All right. Now, so membranes have distinct inside and outside faces. And I'll be talking about, when I was talking about cells, I talked about Golgi apparatus. And I told you that the Golgi apparatus, which is what you have here, you have the Golgi apparatus here. I told you it has the cis side and the trans side. I told you that the cis side is a portion where it receives resources, it receives the membrane, and that's called cis side. Then we have the trans side, which is the point where it transfer or transport its product. All right. So the composition and distribution of protein, lipids, and active carbohydrates is asymmetric across the membrane. Let's go. Now, I've told you in essence, all of the things we have talked about. The reason we talk about the protein is to tell you that the cell membrane is selectively permeable. Selectively permeable means that it determines it is selective. It does not allow everything to pass through it. It is very selective to what goes in and what goes out of the membrane. And that is the reason for the protein. The reason for the protein is because the membrane is selective and some things will not be able to go in, even though they are very needed for the cell, even though they are very important for the cell to use. But because the cell is selective, it's not going to allow them to go in. And because of that, the proteins are there to help transport them. So that is the purpose of the protein on the cell membrane. All right, now the hydrophobic portion of the cell membrane, obviously, because it's hydrophobic, it hates water, so it's going to dissolve in lipid bilayer, and pass through the cell membrane. Hydrophobic molecules rather, they will dissolve in the lipid bilayer and pass through the cell membrane rapidly. So, for example, you have hydrocarbons, they can pass through the cell membrane. CO2 can pass through the cell membrane. Oxygen can pass through the cell membrane. First is this: these are small molecules and they also don't have charges, so they can pass through the cell membrane. But the hydrophobic interior of the membrane will not allow hydrophilic substances, things that has to do like water. That will not be allowed into the cell membrane. So sugar will not be allowed into the membrane. Yes, the orientation is hydrophilic and it's also large. Water will not be allowed in the cell membrane. Ions cannot pass through the cell membrane also. Now, you see, it says that it can pass through it slowly. Now, that's the exception when we talk about water, but we get it. All right, these are proteins that help with transportation. These are proteins that help with transportation. We have channel protein and carrier protein. Now, channel proteins are like channel. They are open. You see, this is open. There's nothing that depicts closing up at any point on it. It is called channel protein. Then there's the other kind of protein that we call carrier protein. The carrier protein, it's not always open. It carries and delivers it. Now, so what that means is that because it carries and delivers, it opens to carry and then close. You see, it's closed here, then open to deliver. And that's the way they work. So sometimes they are closed. All right, let's go. Now, aquaporin is a guide that helps to transport water molecule. Water is able to diffuse through the cell membrane, but very minute quantity. Then aquaporins help to increase the rate of diffusion. So it's also a protein that helps water molecules to enter into the cell. So that's why it's called aquaporins. So they are composed of 4 polypeptide subunits that each form a channel for the passage of water. Overall, up to 3 billion water molecules pass through per cell second. Now, transport protein move only specific substances. For example, glucose carrier protein only transport glucose. So a transport protein that carries water now, it's supposed to carry water alone because it's specific. Proteins are specific in their actions, just the same way hormones are specific. And obviously, most of them are proteins. So a transporter that carries glucose, for instance, is not going to carry fructose, even though they are all monosaccharides. They will not carry different substances. They are specialized to carry substances that they are peculiarly built for. All right. Now, for molecules that does not need protein, that does not need any help to go through the cell membrane, those molecules are able to go through the cell membrane, and the way they go through the cell membrane is called diffusion. Now, so diffusion simply means the process where some molecules are able to transport themselves, move through the cell membrane by themselves without any help. They don't require a protein, they don't require energy, so they just go by themselves. It's the same thing you want to think about when you spray a cologne. If you spray a cologne, for instance, now, once you have a cologne with you and you spray it, if you press on it, you don't need to, like now, if I spray this now, I'm going to spray this to this room now. If I spray this, I don't need, let's say you are in this room now, you are going to, in a minute or two, you are going to start perceiving the smell. Now I can smell it. I don't need to do anything to let it circulate. It will normally circulate into the room by itself. This is what we call diffusion. Diffusion does not require energy. It's spread out, move by itself without energy requirement. So diffusion is the movement of particles so that they can spread out evenly. Now, what is key about diffusion? I will show you on the image here. Look at this. Now, when you look at this, here is water inside this chamber or inside this container, and you see some molecules of dye there. In diffusion, water doesn't move. It is the solute that moves. In a solution, there is solute and there's solvent. The solvent is the liquid that helps to dissolve. The solute is what you are dissolving. So in diffusion, It is the solute that move. Take note, it is not the solvent, it is not water, because water is an example of solvent. So here is a dye that is going to move so that this both area will attain equilibrium. So the dye is going to move, the water will not move. So the dye is going to move from this area where it is high to where it is low, and that is diffusion. The reason it does not require energy is because it's moving along with gravity. It's moving from where concentration gravity is, or concentration gradient. It's moving from where it is high to where it is low. So this dye is going to move from where it is high to where it is low. So it's going to move from here to this side. It's going to move to the right. Now it keeps moving to the right. You see, it keeps moving to the right. It keeps moving to the right until the time that we say it is now at equilibrium. And that is diffusion for you. Diffusion is the movement of solute from a region of higher concentration to a region of lower concentration down the concentration gradient. That is diffusion. Now, so we say this is net diffusion when the movement, when the movement in one direction is now equal to the other direction, we call it equilibrium. At this point, everything is at equilibrium, is equal. Now, this kind of transportation that does not require energy is called passive transport. It is passive transport because it does not require ATP. All right? No energy is required, no protein is required at this point. Now, the other kind of transport, I told you that osmosis only transports solutes. The other kind of transport does not transport solute. It is passive, just the same way as diffusion. And sometimes I like to call it diffusion of water. And in quotes, diffusion of solvents. I told you diffusion, what moves in diffusion is solute. What moves in osmosis is solvent. So here, water is going to move. So osmosis is the movement of water from the from the region where you have more water to the region where you have less water. So in quotes, is a region of, you see, free water molecule. I'm going to underline this for you to see. You see, free water molecules will diffuse across the membrane from region of low solute concentration. Low solute concentration means you are going to have more solvents. It means you are going to have more water. Water is a universal solvent. That's why I use water. So it means you are going to have more water there. And then water is going to move from where it is more to where it is low. So in quotes, it is still passive because it does not require energy, because it's going from where it is low to where it is high. I mean, from where it is high to where it is low. So in quotes, don't forget, even though this is low solute concentration, but solute is not moving here. What is moving here is water molecule or solvent. So since solvent is moving, if it is low solute, then it is high solvent. All right, so you see water is going to keep moving until equilibrium is reached. And so if you look at this area now, look at this. On this area, I'm going to label this A, and I'll label this B. So on the A, you notice that from this side, there's less sugar molecule there, and there's a lot of sugar molecule on the B side. What can happen? If it were to be diffusion, sugar molecule will move from B to A. But in osmosis, sugar molecule cannot move. Why can sugar molecule not move? Because this area, because the membrane is selectively permeable. So water is the only thing that can move. So what's going to happen? Water is going to move from here, where water is more compared to solute. Here, water is less compared to solute. So this is going to, water is going to move from here. Excuse me. Water is going to move from here to B. And that's what you notice that happened here. Here, the volume has reduced. Here, the volume has increased because of the inflow of water. Now, what is going to happen? The parity of this is that by the time you taste this water in A and you taste the water in B now, they will taste equally. They will taste the same. At this point, B is going to be sweeter than A. But at this point, A and B will taste both the same. All right. So this is just a follow-up on that with them. Now, when you say tonicity, what are you talking about? Tonicity is the same thing as concentration. Tonicity is the same as concentration. So it's just like I'm saying, like now I'm drinking coffee, right? And I have some coffee there. I have some milk and some sugar. So if it is not bitter because I've put a lot of milk in it. Now that means if I'm comparing the quantity of sugar compared to the milk, compared to the coffee, I would say it is very concentrated because it's sweet. But if it was the one that is pure coffee, I would say, now I'm looking at the sugar. I'm not looking at the coffee itself. All right. So looking at it now, the coffee is sweet because it has a lot of milk. So it has high concentration of milk. But if it's the one that I didn't put milk, I would say it has low concentration of milk. So concentration is the same thing as tonicity. All right. So you see, tonicity is the ability of a surrounding solution to cause a cell to gain or lose water, which I'm going to be talking about. Tonicity depends on the concentration of the solutes. So when you are talking of tonicity, you are looking at the concentration of the solutes, all right, in the solution. So if you are looking at sugar and water, for instance, now sugar is a solute, water is a solvent, all right. Now, if the solution has a higher concentration of this solute than the inside of the cell, water will tend to leave the cell and vice versa. Now, what we are saying in essence is this: water will always move from where it is high to where water is low. Water will not move from low to high in this case. Now, let's go and look at the scenario. So if you look at this now, for instance, in a case when you have a solution, look at this. I will go back to this slide and show you. At this point here, these two slides. Now, please follow this with me very carefully, please. At this point here, I'll label this A and then B. So here is A. Oops. Here is A. And here is B. I told you the other time that B is going to be sweeter than A because B have more sugar. B is hypertonic. It has high concentration of sugar. A is hypotonic, low concentration of sugar. So A is going to be an you see, low concentration. A is hypotonic, and B is hypertonic. Now, what is the purpose of osmosis? The purpose of osmosis is to make both of them to become equilibrium in the case when both of them will now become isotonic. Isotonic means equal concentration. So the purpose of osmosis is simply to produce equal concentration in both of them. And that's why water moves, so that there will be equal concentration. Now see this here now. So what does this imply to you? Water is going to always move from where water is more to where water is low. That is the rule. Now look at this. So a solution is isotonic if its solute concentration is the same as that inside the cell. So think about it now. If you put my red blood cell, for instance, now some of you that work in the hospital, when you say you want to give somebody drip, what do you give the person? You give the person normal saline, and normal saline is about 0.9%, right, of salt solution. Now, when you put that in my red blood cell is going to be fine if you put that in my system, because that is what the environment is. That is isotonic solution. Now, in that case, the movement of water is the same. There's no net movement. The water is moving well because it is the ideal environment for me, for myself. Now, if you now put my red blood cell in quotes, let's say I come to your clinic and you decide to give me 20% normal cell line, what is going to happen? Because 20% normal cell line have high concentration, it is hypertonic. It has high concentration of solutes in it, of salt. So that is hypertonic for me. What is going to happen to my red blood cell if you put my red blood cell, if you give me such drip? I will suddenly become dehydrated. The red blood cell will become dehydrated. Why? Because the red blood cell will lose water. Water will move from where water is high to where water is low. Now there is more water in the red blood cell because it is in the atmosphere of 0.9% normal cell line. Whereas you have now put it in an atmosphere of 20% normal cell line. So there's more water in the normal cell line, in the red blood cell, than what you have in the normal cell line that you've just injected into my system. What's going to happen? My red blood cell is going to lose water, then the red blood cell will shrink, and then we use the word crenation. We use the word crenation. I'm going to type it here just in case I'm not showing it down. So we say the red blood cell will shrink and then become crenated. So the language is crenation. Crenation. This is close to what happens when you heat your very salty fries. And you notice you've not had water, you just eat your fries, and you notice that you become uncomfortable. It's because you have exposed yourself to a quite salty environment. Let's go. Now, hypotonic is the opposite of hypotonic. Hypotonic is the opposite of hypertonic. It's a state when, let's say, instead of somebody had come to your clinic and instead of giving them 0.9% normal saline, you had given, saline solution, you give them 0% saline solution. So you give them practically water. If you give them water, what's going to happen? Now there's more water in the environment, or let's say you give them 0.1 instead of 0.9. Now you have given them something that will make more water to be there than what is in the cell. What's going to happen? There will be more water running into your cell, red blood cell, and so your red blood cell is going to get more water. And what's going to happen to that red blood cell? It will become inflated and then burst. It will become swollen and then burst. And we call that hemolysis because the red blood cell will be lysed. It's called hemolysis. Now, think about this. If you put this instead of red blood cell, if you put plant cell, it's going to have different experience because plant cell is different from animal cell. Plant cell have central vacuole where they store water. Plant cell have cell wall that is very tough. It's made of cellulose, and cellulose is a polysaccharide that is difficult to digest. And so that makes it difficult to digest. It makes it difficult to digest. And so because, let's say you have put the plant cell in this environment, now what's going to happen? Water is going to go into the cell, yes, but there's central vacuole to hold the water. Now, next thing is this. Then the water is going to still go into the central vacuole, yes, but even if it's now swollen, but the cell wall cannot let it break. The only thing that is going to happen is that the pressure is going to increase. We call it turgid pressure, but it will not break. If you have put plant cell in an hypertonic solution right here, what will happen is that the plant cell will shrink. The cell membrane inside the plant cell will shrink, but the cell wall will remain intact. Cell wall is very tough. That's the issue with plant cell. All right, so this is just what I just analyzed. I just gave to you, showed you the view. In an hypotonic solution, water is going to come into the cell will burst. And we call that hemolysis. In an isotonic solution, it is normal movement of water in and out. In an hypertonic solution, water is going to move out of the cell, and then the cell become shriveled, crenated. And this is what is going to happen if it is plant cell, the plant cell will become turgid, normal. In isotonic solution, excuse me, the plant cell is going to become flaccid here. And in hypertonic solution, the plant cell become plasmolyzed. We call it plasmolysis or plasmolysis. So you see the plasma membrane is lysing from the cell wall. The cell wall remains intact while the plasma membrane lies.